Okay, now let's talk about epigenetics in the brain. So here's my objectives for us. We're going to determine to what degree an ACE score, generally a 4 or higher, can trigger epigenetic processes. And then we're going to predict some epigenetic health outcomes of various ACE exposures. Those are my very succinct objectives for this presentation. And to kind of arc back to the reading, on page 1 of the reading, the very first sentence, in the past 2 decades, the concepts of allostasis and allostatic load have provided researchers with a powerful framework to study both the stress mediators and acute stress exposure and then the damaging effects of long-term stress response. So that is kind of what we're after, is what are the epigenetic outcomes that are part of that. So we can start to define epigenetics maybe at the environmental level, because we've talked about how in human development, a social interaction is so important, and the theory that is probably the strongest argument for that is attachment theory. That attachment, human attachment, is as critical a part of human survival as the hunger response or the stress or the sex response. But environmental epigenetics is a hypothesis saying that environmental events that occur, particularly during development, activate intercellular signals that remodel the epigenome. And we'll define that here in a moment. But these lead to sustained alterations in the structure and function of the genome and thus stable effects of gene transcription. Okay? And if you're wondering where that is happening, it's happening again in that synaptic cleft, particularly around the glucocorticoid receptors. That's the level that we're looking at. Now, we could look at different ways that it affects all sorts of things. That's the one that we're looking at today. Come on in. So I've included now, since I am interested in the imaging side of things, I've included some MRI scans of the brain in the course of development, and I'm looking at from week 1 to 10 years of age. And there's a number of things that stand out to me. These are all taken at roughly the same level on the brain. These are what we call axial images. So if you imagine the brain being like a loaf of bread, we've got slices that progress through the loaf of bread, right? I was always taught by some of the X-ray techs, this kind of looks like a lion's face here. But the significance with this is that we see more gray in the image right here. And so that means that at birth, we have a lot of the brain which is not myelinated. So it has not formed the insulating around the neurons. The neurons are actually still developing, still proliferating. There's proliferation that's occurring even after birth of neurons. So we're growing more neurons, but those neurons are not firing as rapidly as they do once myelination starts to occur. So here we have more white apparent on the image, and that's what that is it's a higher fat signal within the brain. And that higher fat signal is due to the fact that myelination is underway, and these neurons of the brain are insulating themselves in order to conduct signals more rapidly. Right? So that's important, right, for how the brain develops. You can also see that roughly the size of it is increasing over time, and eventually it will reach a size that it will remain, at roughly that size. But we don't see this on this slide, but around the time of somewhere around 12 to 15 years of age, the brain's going to go through what we call synaptic pruning, right? And that's very significant because what's happening is the brain, all of these circuits have been established now, and it's going to say it has some additional wires, if you will, sitting around in the closet. And it's going to say, okay, if they're not wired up by now, just toss them out. So if you can imagine, if we're doing electrical work in a house, we've got a whole lot of electrical wire that we're going to use because we've got to wire up this whole house. So we keep wiring up the house, and we get to a point where we've got everything wired up the way we want it, but we have some extra surplus wires just sitting around. They're not conducting a signal or anything like that. We're going to prune those back. Now that's important for a lot of things, like we see, for example, a lot of people, epilepsy, who have a tendency towards seizures. that's when their first seizure is going to occur. It's around the time that pruning begins, right? Behavior problems, particularly for young men, we're going to start to see those crop up around the 12 to 15 years of age. And again, it goes back to this pruning process. So I want us to be aware of that. And then the final thing, that in terms of the way that the ACE survey thinks about the brain, the initial survey was set to 18 years of age. It says at or before 18 years of age, did you experience this, right? And so that's important that they recognize, these early researchers recognize that brain development was a significant part of how ACEs affected people's lives in a long-term way, but those first 18 years are critical. I would extend the number out to age 25, right? That age 25 is really a critical year where the brain is kind of now fully formed, and all that would change now is just synapses between various neurons. So let's look a little bit about what we're talking about in terms of the sources of these toxic stresses. So I've indicated to y'all already that what we're talking about is an epigenetic process that affects brain development. But sometimes it's helpful to know, okay, well, where is this stress coming from, right? Because there's a tendency when I talk to people about stress, they just think, oh, I'm really stressed about this test, or I'm really stressed about this, that, or the other. And there's always some pushback that, like, well, stress is just a good thing. It helps you, grow up and learn things and stuff like that. That is true. There is such a thing as healthy stress, right? And healthy stress are things that are where the developing child has a caregiver or someone who's going to help them mediate that stressful occurrence. And I mean even things like losing a parent could be a healthy form of stress if there is a caring person there that's going to help them mediate that loss, right? Toxic stress is things that happen recurrently over and over and over again, and there's no caring person there, parent or foster parent or caregiver, who's helping that child mediate that stressful thing. And I am going to differentiate between this type of stress as being in young children. We can talk about post-traumatic stress syndromes and things like that, but that largely, most of that research is looking at people who have had a stressful event either after the age of 18 or the age of 25, as adults, maybe in the military or something like that. That's a different kind of mechanism. It has the potential to have some similarities, but the developmental aspect is important to us. So what we have is, this is U.S. children, and these are just kind of quantified here. This is maltreatment. A lot of times when we talk about stress or abuse, people tend to think about things like sexual abuse or physical abuse. That's actually the smallest number. That's 75 per 1,000 kids age 2 to 5. Postpartum depression has roughly twice that, right? But the single biggest contributor right here is neglect. And it's because of things like the opioid epidemic or parental substance abuse. So, and in foster care circles, we think about this quite a bit, because a lot of times when we talk about abuse, people immediately go to physical abuse or emotional abuse. To a child, that communicates, this person doesn't like me, right? And a lot of times they're able to mediate that by finding someone who does like them, even if it's a friend. Over here in the neglect category, it's like if mom or dad and there's no shame here. I don't want you to hear me talking about shame. I'm just talking about situations that people wind up in. If we have a caregiver who's not responsive because they're abusing drugs, that neglect communicates to the child, I do not exist, right? And in terms of what we talk about, the dyad of interrelationships between a parent or a caregiver and a child, that has a more pronounced toxic and long-lasting effect. So let's think about these different types of stress and how the stress response occurs. So we've talked about how there's positive, tolerable, and toxic stress. Positive stresses are things like stressed out before a track meet. I'm going to run better that day, right? Tolerable is literally, we would think it's really bad situation, like parent dies, but my granddaddy is there to help me walk through it. I'm going to call that tolerable. Toxic, I love Brene Brown's example of what toxic stress is. So she says that the stress response, again, is, the cave bear is chasing you, right, and you get away from the cave bear. Toxic stress is, imagine the cave bear comes home every night, right, and you're living in the same house with the cave bear, and you don't know what that bear is going to do. So we said that the system is meant to be an acute response system, and it mediates a lot of different systems in the body. It increases blood glucose, so it provides more energy for the cells. It increases blood pressure. It gets more blood out, more oxygen out. It modulates immune response because that burns energy, so it will suppress immune responses, or it will change immune responses, and that's somewhat variable. It reduces motivation for rewarding stimuli. So again, that goes back to the hunger and the sex drive. Like if I'm being chased by a saber-toothed tiger and I see an advertisement for a hot dog, I don't need to be thinking about hot dogs right now, right? So I'm not thinking about how great it would be to have a hot dog. It increases vigilance and arousal, and the ways that we think about that as arousal is whether if you flick someone on the ear, do they perk up? Right, that's what we mean by arousal in the sense it's awakened state, right? So again, if saber-toothed tiger is chasing me, I don't need to think about, oh, I'm so tired right now, I just want to take a nap, right? I need to be awake and oriented to my environment. And then we said that finally, through the hippocampus, it's going to do a consolidation of memory task where it reminds me, okay, by that hot dog advertisement, that's where the saber-toothed tiger was, right? So avoid the hot dog advertisement. Now, that's an acute stress response that's very helpful. But over here we have this affective persistent stress, and this is kind of where we're at when we think about the neurological part. We're over here on this side. So I know that we've already probably talked quite a bit about stress, but I wanted to bring it back because what we're dealing with is how does this persistent process occur? And the persistent process is largely because of, again, the way that the hypothalamus and the pituitary gland are working to keep a chronic stress level. And so this is things like excessive, now blood glucose, excessive blood glucose, and we have type 2 diabetes, right? Hypertension, because we've increased the blood pressure over weeks and months. Now we've got people who have heart problems, right? This one I think is one of the saddest ones, but modulation of immune response, vulnerability to inflammatory diseases. So that's things, again, like autoimmune disorders. And when I think about particularly how many women in this country suffer from autoimmune disorders, and if that is part of the way that our society or the way that women are experiencing stress, that's really sad if you think about it, and we need to own that. Loss of interest. So if I don't care about hot dogs at all, right? Which I don't really because I'm more or less vegetarian. But say I've just lost all interest in anything fun. Fun is overrated. I just want to sit in my bedroom and listen to goth music, which was me as a teenager. Then we have depression, right? Okay. I think in particular about young men and women that I see who have headphones on all the time and they're listening to their music very, very loud. And if you really sat down and tried to figure out why are you listening to your music so loud, well, can they even hear it anymore? Or is just their lives so stressful that they need that volume turned up that much more to help them maintain their interest? Does that make sense? Again, me as a teenager. Hyperarousal and anxiety disorder. So it's good to be awake if something's trying to kill me. It's not good to be awake for 3 nights on end, right? Because, I'm worried about so-and-so winning the election or whatever. And then preponderance of adverse memories. And these are perhaps in some ways the most toxic because if we're dealing with caregivers and mother and child relationships or father-child relationships, most of the research looks at mother-child relationships. If we have a mom that is triggered by her child, it's triggering aversive memories because she's remembering how she was abused when she looks at her child. And the child, when he looks at the mother, is thinking about how mama got beat up last weekend or whatever. She triggers aversive memories. Now we're just feeding off of each other, right? So this is a real. This one doesn't sound too bad, but it's actually maybe the worst of the bunch in some ways. And again, these are being mediated by the brain. So I'm not going to spend a lot of time on this slide, but I just want to say that a lot of what I'm saying is grounded in the ACE study, and that there is some, we can't see it real clearly on this, but if you see this little guy right here that's highlighted on this slide, that's what I was trying to point at here in this really blurry, terrible slide. Okay, and what we says is that it says the ACE study revealed that individuals with a high degree of ACEs exhibited greater than twofold elevation and perception of current stress than those who did not have such a high ACE score. So people, if they had a high ACE score, they are more stressed out as a rule, right? And if we zoom in over here, I'm going to zoom in. So what we're talking about is the amygdala. That's what we're seeing, right? Is activation of the amygdala, this emotional center, right? And so if I slide over to this chart, oh my goodness, this is terrible. But what they were doing, this is completely illegible. They were doing MRIs of people and showing them pictures of people's faces. And they were seeing what amygdala activity occurred when they saw different kinds of faces. Like if they saw a happy face, an angry face, a fearful face. So this first one is angry face. We see over the comparisons in black, we have an increased activation of the amygdala in response to an angry face, right? This is a fearful face here. So, typically developing child, right? High ACE score child over here. Elevated amygdala in response. So they are internalizing the stress, even just of an image. Like they're seeing someone who looks afraid, and they're piling it into their own network, if you will, right? And this is interesting. This is where it gets really sad. So we said this is typically developing. Oh, wait, typically developing excelled them. Can anyone guess what this face is here? It's the happy face. When they showed them the happy face, it just makes me want to cry. The amygdala is less activated in the child who experiences toxic stress. They have less of a response to a good thing, right? So not only did it heighten arousal around fear and anger, because that's what's going to keep you alive. That's what the lizard brain is looking for, the fight, flight, or freeze response. It has a decreased response to positive social input. That again goes back to that the thing like, just don't care anymore, I'm just going to turn my music up. I don't even see that people are smiling anymore, right? I always think about the old quote, like, laugh and the whole world laughs with you, cry when you cry alone. This is it in science terms, right? And then finally, this is neutral faces. So just people with no affect on their face. You see some response among the typically developing, and then the children with known ACEs, less of a response to just neutral faces. So they're not looking for that, is what this is saying. They are looking for what do I need to run from, and what's going to try to kill me, right? If that makes sense. That's what the amygdala is primed for now. So here is, this is controversial data here, but I did want to share it with you. So this is talking about the hippocampus and prefrontal cortex. So how we move from memory consolidation, we said the hippocampus is going to help us consolidate memory. So I'll remember, okay, that's where I got chased by that saber-toothed tiger, right? And where that's stored in the prefrontal cortex, where that stressful response is stored. And what they have found, and this is somewhat controversial, is that there may be reduced volumes of both the hippocampus and the prefrontal cortex in folks who have increased stress response. That the stress system is now starting to impact brain volume and impact myelination. So that we have now, and this is really, really important for you to hear me say, there's no judgment in me when I'm saying that. I'm not saying that these people are dumb or something like that. What I'm saying is that their brain is now ideally suited to survive in the environment that they're in. Right? So what the brain does is it is built for survival. I mean, sometimes I think about it as kind of a dreamy, fun place. I like having imagination and coloring and stuff like that. So that's one way to think about the brain. But the reality is the brain is a machine built for survival, just the same as your heart, just the same as your kidneys. It is built to survive. And so when we see it start to change volumes or change myelination patterns due to stress, what we see is we see a machine that's surviving with what it has, the inputs that it has. So what this is arguing is that reduced volume of the hippocampus of prefrontal cortex may contribute to, sensitization of stress response due to the loss of inhibitory roles. That's a bunch of fancy stuff for saying that it is impairing the development of language and logic centers while it is enhancing the power of emotion and memory consolidation centers. So we have someone who has an intense emotional intelligence, but maybe not all of the intelligence for expressing themselves or for thinking about future consequences, right? So this is the person, and you may know some folks like this. This is the person who can read a face really quick, and they know what that person's thinking about, right? But they cannot think about next week. They're not thinking about, where the rent's coming from or something like that, okay? Questions about that? I think about that because when I was doing my master's degree, I was living in a rough part of town in a different city. And I got called professor a lot. There was a lot of prostitutes in this part of town. There was a lot of rough characters in this part of town where I was living because it was graduate school. Which graduate school could be better than that, but that's where I went to graduate school. And they called me professor a lot. They would give me a lot of grief, right? Because I could not figure out situations that they understood very quickly. Like, they figured out these situations really fast, and it would be like a week later, like, oh, that was going on, But then they would be coming to me and asking for rent money, right? So there's that tension again. So this is very real stuff to me. This isn't academic.
Are you talking about like street sense or something?
Yes, street smarts. Like, if you ever heard someone say, like, I live by the streets, I'll die by the streets. Those kinds of things. Or like I had one guy tell me, the streets are my mother. Right? She's taught me everything I need to know. And there's a lot of truth in that. Like, I'm not discrediting that truth. That's an important truth. I would love it if we could find a way to have both. Right? That's a really good way to frame it. Thank you. So let's zoom in yet again and look at what's happening at the neuron level. So these are neurons. I believe that this study, they looked at monkeys, so other primates. And this word shrinkage sometimes gets applied to the actual neuron itself, right? And so what we see is a pruning of synaptic connections, right, among certain neurons. So it's just base level versus chronic stress situations. So what we're saying, it can happen at kind of the global level of the amygdala. It can actually happen also at the cellular level. So how is this occurring? Well, both the prefrontal cortex and the hippocampus are involved in stress perception because they're working with consolidation of memory, right? And if they're impaired in the way that they're thinking through this stuff, we have a vicious cycle that the person enters into. Where what happened initially as a feedback loop that was meant to tell the system to turn off becomes a feedback loop that just reinforces the system. So y'all remember when we drew the feedback loop on our thing? What I'm talking about is a feedback loop that's now being triggered by memories. So I'm remembering this terrible thing that happened when so-and-so came home and they did that terrible thing. Oh, they're coming over this weekend, right? And so I start now initiating this vicious cycle. So stress arguably decreases hippocampus and prefrontal cortex, which leads to an increased perception of stress and an impaired way to terminate that stress response, which then leads to more stress, right? So we just have now this vicious cycle of a person cycling through heightened states of stress. So let's talk about the role that glucocorticoids play in that, right? They are the part that is the feedback loop. They're meant to terminate the stress response. They are the stop. They're the brakes, okay? So if you look on your list of key terms, the way that I've defined glucocorticoids is that they're an important component of the stress response. They help redistribute energy resources, and then they also serve to stop the stress response. And the fancy way to talk about that is they work to restore homeostasis after the end of stress. Okay, so once the challenge has ended, the glucocorticoids, the brain picks up, okay, there's glucocorticoids floating around, I can relax. I can take a deep breath. I can think about that hot dog that I wanted so bad. Right? I can turn my music down. Of these, cortisol is the most important. And if we're going to try to make a connection to clinical applications, that's one. So I know we just spent a lot of time talking about a lot of stuff to finally get it a clinical application. I'll try to sprinkle more through. But we can actually measure a child's cortisol level and know how stressed that individual is, right? And it's very helpful because sometimes a child may have learned behaviors where their affect is very neutral and they don't appear to be stressed at all. But then when you take a sample of their blood cortisol level, it's through the roof. They've just learned how to have a behavior or an affect that masks the panic that they're feeling. So, evidence suggests that early life stress results in a state of glucocorticoid resistance. That's what we're driving to. How do we get to an individual that resists the message to turn off the stress response system, this HPA axis? They have the axis working, but it doesn't stop. So that we would call that glucocorticoid resistance, right? And we're just going to define that as an impairment of the actions of the glucocorticoids, which are there to largely to help terminate the stress response system. I don't want to simplify things too much. I am a little bit. This glucocorticoid resistance is a major component of this theory of how early life stress produces long-term health outcomes, right? So how a person who had an ACE score of 4 or more is now going to have an autoimmune disorder or heart problems. What we've done is we've affected something at the basement level of the house that's now affecting the whole building, right? And this regulation of inflammatory processes by glucocorticoids is the key to understanding the effects of glucocorticoid resistance. So we're going to look at that when we look at epigenetics and we talk more about epigenetics. Let's talk a little bit about glucocorticoids and immunity, though. So they act on white blood cells to reduce inflammatory molecules, right? And this helps to limit inflammatory responses caused by stress. And that's good because if I'm running for my life, my body does not necessarily need to be working to fight off infection. That's just burned energy that I need right now. So resistance to the action of glucocorticoids on white blood cells results in an increased systematic levels of inflammation. It's kind of counterintuitive if you think about it. But since it's meant to temporarily limit the inflammatory response, if we keep it on for too long, the inflammatory response is eventually going to take over it, right? It's almost like it's pushed down, and then just springs back, right? And that's one way to think about maybe even how glucocorticoid works, and these processes work on depression. You push the thing down until ultimately you're just depressed, right? It's a similar process in very simple terms. All right. To fully understand this slide, we are going to need to file this one away. But this is an important slide. It's giving us something very, very simple. That I think this comes from a mouse study or a rat study. But what they did was they, or maybe this was monkeys. I don't remember which. They took animals, right? And they would raise the pups with the mom, and then they would remove the pup from the mom, right? And they were looking at what happened at the synaptic level with children where that happened at different levels of development. Like, for example, week 2 after birth versus 2 months after birth or 3 months after birth. They looked at different developmental milestones, right? And what happened. So in a standard developmental condition, we have this glucocorticoid receptor gene, right? That when it's activated, it makes glucocorticoid receptors there in that synaptic cleft. And we have a pathway that's allowing for activation of production. So I'm now at the genetic level. I'm saying that inside the body, you have DNA. And if you think about it, every single cell in your body has DNA to produce the entire body. So what I'm saying is, it's a funny thing, but if you think about it, the cells that are in your eyes have all the information to make your ears. So why don't we grow ears out of our eyes? Right? You get what I'm saying? They have all the information there. All that DNA is there. What it doesn't have is things that are turning on the protein synthesis. So it turns off the cells in your eye, turn off the parts of the DNA that code for your ears. We're going to call that an epigenetic process. Right? And we'll look more at that in just a sec. But what I'm telling you right now is that in standard developmental conditions, the DNA, when it's synthesizing proteins for glucocorticoid receptors, it synthesizes them. It makes them. If we have early life stress at different levels of development, what we have are chemical markers, they're called methyl groups, that tell the DNA don't code for that. Don't code for those receptors. So we have a decreased number of glucocorticoid receptors that are being produced. Right? The brain is saying, we don't need those. We need to be stressed. Right? So that's what we're driving at. We will come back to that slide, though, but I wanted to go ahead and bring it in now. So in review, and I'm sorry, some of this got cut off. These ACEs that we've talked about are going to increase the epigenetic markers that are there on the DNA that silence glucocorticoid receptor production. Okay? And this reduction in glucocorticoid receptors results in developmental glucocorticoid resistance, which in turn heightens sensitivity to stress, impairs feedback, and increases inflammatory pathways. Right? And finally, this shift in function increases susceptibility to mental illnesses, especially depression, as well as conditions like obesity, heart problems, autoimmune disorders. It just kind of goes on and on. Everything else that the ACE study indicates. Okay? Does that make sense so far? Okay. Anytime we have increases sensitivity to stress response, so a person perceives stress even when there's not stress. And I think about this particularly with young adults. This alters function and structures of certain parts of the brain, particularly this the prefrontal cortex and the hippocampus. This is still, like I said, somewhat controversial, but definitely the prefrontal cortex. No one debates this. There's been different findings about the hippocampus. Impairments of how the brain function or changes in how the brain functions further disrupt the stress response system and cause basically a syndrome of prolonged stress. And these impairments to the prefrontal cortex, to the language and logic system, contribute to increased risk-taking, impulsive behavior such as smoking, drug use, promiscuity. Right? So this is speaking more to sometimes what we talk about the social aspects of ACEs. But I am arguing that this has a neurological way of thinking about it as well. And that's it for this presentation.